Hello, my name is Tomaž Skrbinšek, and today I'll be talking about how we use citizen science and genetics for brown bear monitoring and how we do this in Slovenia. Uh, I come from University of Ljubljana, from biotechnical faculty, from Slovenia, uh, but where I'm a lecturer and researcher, and I'm also one of the co-founders of the DivjaLabs Wildlife Genetics Lab, which is a university spin-out company. This lecture was originally given at Purdue University in September 2025, where I gave it as an invited lecture, where I was invited by Professor Andrew DeWoody. Um, the lecture was supposed to be recorded, but something went wrong technically, and since there was a lot of interest in the lecture, I was asked to re-record it. And well, this is what you're seeing now. Okay, to get back to the lecture, first let me tell something about Slovenia, about the country where this is happening. Our country lies in the south of Europe, at the edge of the Mediterranean Sea. You can see it in the middle map there. It's a very small country. It's like a small dot on the map. Just with the sense of scale, I've put an outline of our country over the outline of Indiana, so you get an idea how small of a country we really are. However, for such a small country, we're very diverse. We lie at the intersection of four large European ecoregions or biogeographical regions. We have the Alps to the northwest, we have the Mediterranean Sea to the south, we have Pannonian Plains to the northeast, and then we have the Dinaric Mountains to the southeast. The Dinaric Mountains are a very rugged area covered with dense forests, and this is where we have most of our large carnivores. One of our large carnivores is the species I will be talking about today. That's the brown bear. European brown bear is a very close relative to North American grizzly. It's the same species, a different subspecies. It's a bit smaller, maybe a bit less aggressive than the North American grizzly, but it's still a very big and a powerful animal. Like most large carnivores, it gets in trouble with humans and human interests, and this makes its conservation, a challenge. The problem for conservation of brown bears, it's not the brown bears, it's the world they live in. This satellite photo of Europe at night tells everything. You can see which species is dominant here. Like modern day Europe, it's 700 million people, millions of kilometers of roads, agricultural areas, industrial areas, urban landscapes. There's really not any wilderness left. We don't have any large wilderness areas where we could keep these large carnivores. Whatever wants to survive in this landscape needs to survive in coexistence with humans. This is quite different to the separation paradigm which is used in North America, in Africa, where large carnivores are separated from humans into wildlife reserves, into national parks. In Europe, if we want to do that, we wouldn't have any large carnivores left. They just need two large areas. So we have to work within this coexistence paradigm. To make things worse, Europe is also heavily politically fragmented. There's a lot of countries, and also wildlife management and conservation is often not even done at the level of a country. It's often done at the level of a region or a province. Which makes this management fragmentation a real issue and an additional challenge. However, we're doing quite well. In a now not so recent review, we showed that European large carnivores are really doing okay. All populations, or the vast majority of populations, are increasing in size, increasing in range, and over the last decade since we published this, things have even improved. So we are quite good at implementing this coexistence paradigm. But then again, of course, this increase of abundance and range of large carnivore brings conflicts. And actually fixing these conflicts and ensuring long-term survival of these animals in our landscape requires a lot of knowledge. Okay, to get back to Slovenia and to our brown bears, Slovenian brown bears are the most northwestern part of the Dinaric Pindos brown bear population, which is a large indigenous bear population that stretches along the Adriatic coast from the Alps to Greece. Although we actually take only a small part of the entire population range, the bears in Slovenia are pretty important, since we are any natural connection between the Dinaric Mountains and the Alps, where we would like brown bear recolonization to happen, has to go through Slovenia. Also, bears in Slovenia were the source of all brown bear reintroductions in Europe. Bears here were not always doing so well. At the beginning of the 20th century, they came quite close to extinction. Maybe only about 40 animals estimated remaining. The situation improved and the population recovered, and then some protection, at least through cull quotas and some management, was started in the 1950s and 1960s, first by hunters, the hunting organization, and then later on also by government. The population recovered through 70s, 80s, and the first half of the 90s, but then the problem started. Actually, towards the end of the 90s and the beginning of the 2000s, the bears became more and more of an issue, and there was more and more of a talk about that, and came into the focus of the public. Of course, when things were already heating up, bears, being bears, tend to do something stupid. This is one case that was really heavily present in the media, where it's have blood and snow. What's better for a journalist? This guy got mauled by a bear. He got a couple of stitches. The bear didn't fare that well. However, it was also put some more kerosene on the fire, and made the whole debate even hotter. All this combined and the increase of this human-bear conflict brought about the increase in cull quotas. In Slovenia, we hunt bears, always have, but it's strictly regulated. There are hunt quotas, and people stick to that. But these cull quotas basically doubled overnight, and there was a lot of questions about sustainability of this practice, both from experts in the country and abroad, and also by the politicians in the European Union. And some of these questions were pretty unpleasant. But the core of the problem was that we really didn't know how many bears we had. Estimating wildlife abundance, out in nature is really hard. You cannot, tell the bears to stand in line so you can count them. You can count yourself lucky if you even see them when you go in the forest. So you're always limited to some sort of indirect methods. Biologists have been for the last 100+ years using capture, mark, recapture. So the idea is you go out, you capture animals, you mark them, you release them, then you go out again and then you capture them again, and then some of them are marked from before. And then from this ratio between marked and unmarked animals, you can use, a bit more sophisticated mathematical modeling than that, but you can actually estimate the total population size, including the animals you never captured and marked. So of course the critical part here is marking the animals. And biologists are pretty creative, we tag them, we ring them, we draw on them, put colors on them, all sorts of stuff. The problem is, if you want to do this with brown bears, capturing, tagging, releasing, recapturing, that's really not going to happen. So these methods were pretty useless for brown bears, for a long time. All changed with rapid development of molecular genetics. Now the recipe is pretty standard. We use non-invasive genetic sampling. So we take the genetic material the animals live in the environment, for example, scats, urine in snow, hair, etc. And we genotype those, meaning like we would take a genetic fingerprint. So we individually identify the animal. So when we first capture an animal, find its sample, we can call it a capture and marking. Whenever we get the sample of the same animal, either being this a non-invasive sample, a scat, a hair, or tissue of the dead animal, we know it's the same animal, and we can call this recapture. Put together, we get this capture, mark, recapture concept, and we can estimate the population size. So this is how our bear monitoring evolved in Slovenia. We started already over 20 years ago as a pilot study in 2004. This was my PhD project. We really didn't know what to do back then. We were just trying to see if we can do this at all. I got a lot of help at the time from Professor Lisette Waits from University of Idaho. And I'm really thankful to her for that, because this really helped, to put us in place and kick us in gear and actually get this going. Now, this pilot study was a success, so this gave us confidence to start in large-scale studies, and we did the first large-scale, the entire country population estimate in 2007. In 2015, Croatia joined in, and our last cycle was in 2023. So now this is becoming pretty much routine, and we have this routine 8-year cycle for genetic census or genetic estimate of the population size. I won't be talking about every study, but I will be describing how this last study in 2023, how we did it and how it turned out. Okay, so first, to walk you a bit through our study design. We did this exercise first in 2007 when we did it for the first time, but it's pretty much stayed the same. So these are our parameters of the study. It's pretty high intensity. So we estimate using market capture simulations, we estimate how many samples we would need to get acceptable statistical power according to the assumed or guessed number of animals in the population. So in our last study, this goal was 2,500 samples. We wanted to keep population closed so that not many animals would move out of the country or move into the country across our national border with Croatia, where this population extends into. But also we didn't want to cross the reproductive cycle, so we didn't want bears to, get born while we were doing this, because this would really mess up our mathematical modeling. So we wanted to do this in 3 to 4 months. Of course, we had limited funds. You never get as much money as you would ideally like to have for stuff like this. And no reruns, meaning we got one shot. If we mess it up, we mess it up. So high intensity and short meant we had to have a lot of people in the field. And because our funds were limited, these people had to be volunteers, or now it's fashionable to call this citizen scientists. So, and because, we had no way to repeat stuff if things went wrong, we really had to do thorough planning and preparing. We couldn't just go and do some sort of a half-assed attempt. We had to do it, and we had to do it right. This is how our sampling network looks like. So we have 2 main sampling networks. One was hunters. Hunting organization in Slovenia is very strong. It's our largest NGO. In our small country, we have about 22,000 hunters, and they're really well organized. And another network that actually covered most of the same landscape were foresters, employees of Slovenia Forest Service. And there are also some professional hunters employed in the Slovenia Forest Service in the hunting reserves run by Slovenia Forest Service. They're marked in green on this map. Apart from that, we had quite some interested individuals, NGOs, which also participated in this. Then one of the goals we want to have is to have equal effort over the entire area, so that not a lot of people, so that the same number of people over the same area would sample all over the entire Bear Range. We tried to solve this so that we actually gave to each participating hunting club or forestry department the amount of sampling material corresponding to the area of forest in the area they covered, which should give us approximately similar effort. It of course meant that we would be giving a lot of material out that would never get samples back in the areas where bears are rare, for example, high in the Alps or to the northeast of the range. But still, this is the only way to get statistically sound sample. Organizing such large-scale citizen science sampling is a huge effort. For example, this was the third time we were doing this, so we knew already what we were doing, and it still took 5 months of work to organize that. And actually, the same amount of funds as we needed for all laboratory work and data analysis and all that. We had to organize a lot of pre-sampling meetings with hunters and with foresters and other volunteers. We had a media campaign, wrote articles in hunting magazines, in daily media, gave interviews. So it was a lot of effort just to get the message out that we're doing that. We really thoughtfully prepared sampling kits so that everybody would have this really professionally prepared, nice looking box with everything they needed for sampling, instructions, material, everything inside, so that we looked and acted professional. We ended up distributing 7,500 sampling kits to the field for this sampling. Another important part of our sampling design or preparation for our sampling was to plan for contingencies. Now these slides are actually from the project meetings in our 2015 study, which we did together with Croatia. And the size of these squares says how many samples we expect from each stakeholder group or from each area. This is plan A, so everything going smoothly. We'll get more samples from hunters, from professional hunters, a bit less from foresters and general public. But this is, if everything went well, this is how we would expect things to look like. But as things don't always go well, this is our plan B. So if and when problems happen. So we were prepared. Then if sampling was not progressing as we would like in a certain area, we would react. We wouldn't wait for the sampling to be over because once it's over you cannot travel back in time and collect more samples. We reacted. Ask where's the problem, alert bosses, depending on where the problem was. In the worst case scenario, we would mobilize paid staff, mobilize some volunteers from general public and actually go there and sample ourselves. So this was the idea. And the key for that is that you need real-time monitoring of sampling. So meaning you need to know how many samples are collected in particular part of the sampling area, so that you can actually detect that there are problems. Okay, so the sampling started in September 2023 and lasted until December of that same year. Most of the samples were collected in 3 months, September, October, and November. It was really high intensity. What actually made this easier is that we had mailing collection of samples. So every participant in their sampling package, they got this pre-addressed postage paid envelope. So when they found the sample, they put it in the envelope, threw it in the first mailbox, and the sample got to us. This allowed us to get samples back close to real time and we immediately entered them into a spatially explicit online database so that we could see how the sampling is progressing, and if there were issues anywhere, we would react. We didn't have issues in this particular study. We did in previous studies, and we did have to react. So this is really, for peace of mind, this is the best thing you can do. The sampling was absolute success. We were aiming for 2,500 samples. We ended up collecting 2,864, plus 62 tissues of bears that were killed during sampling. To get an idea of the scale of this, 907 different people sent samples back to us. In the areas with low population density of brown bears, most people didn't even find a bear sample, even if they were out there with our kids and wanted to help. So much more than that, people actually participated in the study. We captured, quote unquote, 656 different bears. So this is the minimum number of bears we had at the time. So this is how many different genotypes we detected. Our average recapture rate was 2.68, meaning that every animal that we captured was on average captured 2.68 times. So not that many bears actually managed to escape our sampling. If we put this on the map, this is how it looks like. This spaghetti thing here are tracks of animals. So blue are males, red are females, each dot is a sample, and lines connect samples of the same animal in chronological order. So it's kind of a parallel to those of you that are familiar with telemetry. It's kind of parallel to telemetry, just you have small number of points for a lot of animals, which is exactly the opposite what you usually have in telemetry. There's a lot of interesting patterns that emerge when you do that. I'm not going to go into all of that now, but one thing you can see is the effect of Ljubljana-Trieste highway. This cuts the bear range, separates the Dinarics from the Alps. It doesn't have any suitable wildlife crossings. There are some places where animals can, with some danger to themselves, cross, but it's definitely a barrier. And sampling was the same intensity on both sides. Landscape is similar, but on the eastern side of the highway we have most of our population and only a couple of bears to the west. So it is a serious barrier. To get back to the population size estimate, now first I'll describe the graph on the bottom. I really like to show mark recapture data in this manner. I call it the mark recapture saturation graph. So each dot is a sample. Again, blue are males, red are females. And whenever we find a new animal, we make a new line. So each line is an animal, and each dot on that line are samples from the same animal. And so on the left side of the graph, you can actually see the shape of a saturation curve. And as time progressed, time goes from left to right, from beginning of sampling to the end, you can see that there were less and less new animals as things started saturating, and more animals were just getting recaptured. And then the red line on the top is our mark recapture estimate, and the gray ribbon around it is the confidence interval. So you can see the difference between the last animal in the graph and this red line is actually the estimated number of animals we didn't capture. So the minimum yearly size, which we directly estimated, this is after all mortality, excluding the animals that died during the sampling and before that, and corrected for edge effect at the border with Croatia. It's 737 bears with a quite narrow confidence interval. We also derive from that the maximum yearly size, because we shoot a lot of bears every year, and bears die also from other causes. This number really fluctuates over the year. So if we just add recorded mortality, we get to 1,001 bears in spring of that same year. Again, with a relatively small confidence interval. And we got a 60 to 40% females versus males sex ratio. So this t-shirt was photographed in the wilderness of Ljubljana souvenir shops a couple of weeks before we published the results, and the souvenir people already knew, we had 1,000 bears, so we shouldn't have bothered. Anyway, if you do sampling once, that's not monitoring, that's a snapshot. It becomes monitoring once you add the temporal component. If we look at the long-term perspective, we did by now this 3 times, we can see that the population is steadily growing. It's steadily increasing since 2007. On the other hand, what's interesting is that the sex structure, sex ratio is remaining surprisingly stable. It's reflected, this 60 to 40% females versus males actually really well corresponds to the cull, the balance in the mortality. Females with cubs are not being hunted, and generally more males get shot anyway. Although it's not trophy hunting, even the sex structure of the culling is strictly prescribed. But of course, we do this every 8 years, and we need management data every year, right? So what do we do? It would be, at least for our circumstances, completely impossible to do this every year. It's too expensive, it's organizationally too difficult, and frankly, I don't think that even the most enthusiastic volunteers would get sick of us if we ask them to collect bear shit for us every year. So what we do is we make population dynamics models, which we calibrate or anchor with our empirical field estimate of the abundance. This is done by my colleague Klemen Irina from Department of Forestry at our faculty. Now, models are even based on really solid data and calibrated with empirical data. They're a bit like a weather report. They're really good. Like weather report is excellent for tomorrow or the day after tomorrow. It's useful a week from now, but a month from now, it's anybody's guess. So of course, the further away you get from the calibration point, the less reliable your model estimate becomes until it's useless at a certain point. I had my doubts how this was going to work, but actually the model turned out surprisingly well. So just to kind of clarify things, so the model gets recalculated every year with the most recent mortality data. So it gets updated. But if you look, the thick black line with dots in the middle, that's the model predictions, and the gray lines are confidence intervals. And this last model, the one here, was produced in spring 2023, before the new genetics estimate. And the new genetic estimate was produced in November 2024, and it was really, really close to the model prediction. We got really lucky, I would guess. It's not always going to happen like that. This is really close. But nevertheless, it is useful. We can use this for management with quite more confidence now. So we use this as basis for long-term management. Our goal is to have proactive management, not reactive management. Our current management goal is to have 800 bears in spring. This is our estimated social carrying capacity. The idea of this can change in the future, one way or the other, but this is our current goal. And we should, in management, follow this goal based on the best possible information we have. But this is not without challenges. For example, you can see this upward deviations in the curve and downward deviations in the curve. For some years, the culling was completely stopped due to court rulings, and of course, the population went up. And then backlash was after this was cleared. Then this was reduction culling, and then we shot a lot of bears, and the population went down again. We don't, what we really don't want in managing brown bear population is this flip-flop systems where you have periods of full protection, then something bad happens, and then you get this knee-jerk reaction from the politicians, and it goes completely the other way, and you have this bunch of animals getting shot. This is the wrong message to the public, and it just polarizes everything, and just it tells to the public that you really don't know what you're doing. This is right when we published this results. A couple of weeks after that, there was a fatal mauling, a fatal attack of a brown bear on a human in Slovakia, where bears were completely protected, and then this panic reaction by politicians and decreed cull of 350 bears. So this is not how this should be done, and with public pressures also in Slovenia, we may be heading in this direction where we'll be doing these flip-flopping things instead of having proactive management with clear goals and based on data. Okay, so to start wrapping this up, we have been collecting bear shit for the last 20 years, and what we've learned this time, besides that bear shit in the woods, one thing that became really clear is that sample quality is king. Really, researchers should aim, as much as effort as possible to really getting high quality samples. The people that work with us in collecting samples, we always give them instructions how to estimate, subjective age of a cat, and only to collect fresh cats. We take care that they get good materials for collecting samples, because this really makes a difference. But even in the same study, we can get in different areas different success rates. For example, these data here are from Slovenia creation study in 2015. Everything was collected with the same material, the same procedure, analyzed at the same time in the same lab. And still the success rate in Croatia was 66% and in Slovenia 79%, which is quite considerable difference. Now the problem which we identified when we later analyzed the data is that Croats on average collected older samples, which may be the case just because it was what they could find or whatever. This is subjectively estimated age of the cat. In any case, this made a difference and resulted in different genotyping success. So we had with different species, we had genotyping success everything from 60% to over 90%, but it's not always something you can influence, but you should really try to do it as good as possible. Now, laboratory procedures really improved a lot. In 2005, when I started doing this lab work, it took me 6 months to analyze the first 100 samples. In 2024, it took us 6 months to analyze 2,500 samples without even breaking a sweat. We are a relatively small lab, but we could easily or comfortably analyze this number of samples. In 2007, when we did the first time the whole country study, it took us 2 years from sampling to the final result, and we only collected 1,000 samples. There were less bears then also. So 1,000 samples was good enough for a good result, but it took us a long time to process that. In 2023, it took us 10 months from sampling until we gave the report to the managers with 2.5 times more samples than in 2007. This really makes a difference when the results are supposed to be used for management. If you get data 3-4 years later, that's really not useful anymore, or it's a very limited use. So this improvement of laboratory methods is really making this more relevant and more attractive for management. Another thing that really makes a difference for us is that we completely switched to genotyping by sequencing. We're still using microsatellites, which are excellent markers. The problem is that they used to be analyzed with capillary electrophoresis, which is difficult, subjective, and it's plagued with problems. But already in 2015 study, now over 10 years ago, we completely switched to a method where we actually sequence microsatellites on a high throughput sequencer on an Illumina platform. This has numerous advantages. One of the advantages is speed and less manual work, because a lot is done with bioinformatics. If we combine this, which we're doing, with laboratory automation, with robotics, we can get, even in a small lab, really high throughputs. And what's even more important probably is that the data is completely objective at the level of DNA sequence, completely future-proof and completely transferable between laboratories. So this has been a major change for us, and we've shifted to this for all other wildlife species we work with. One other thing that I also like to point out when showing this is that laboratory is easy. That's the easy part of the study. I'm not saying that it's simple. It takes a while, and it takes a lot of expertise, to get the methods established in the lab. It takes a lot of care to do things right. But things are under your control. It's really difficult to mess up catastrophically once the samples are in the lab. You would really have to do something terribly stupid. On the other hand, getting samples is hard. The fieldwork part is really difficult. Many things are not under your control. Many things are unpredictable. And in a lab, if sample doesn't work, you can repeat it. In the field, if the sampling doesn't work, you cannot go back in time and repeat it. So it's really hard. We had our share of failures with other species in other areas. We do wildlife monitoring also on other species, and not only in Slovenia. Nobody likes to talk about failures, but in all cases the problem was in the field. Sometimes it can be just purely bad luck, but often it can be people not taking that part seriously, not understanding how challenging that really is. They see the genetics as this big, complicated thing, which it really isn't, and underestimate the effort needed in the field. So that's something I really like to point out. The field part is hard. Another thing that I like to point out is that if you're working with volunteers, if you're using citizen science, you should make participation as simple as possible. You're asking people to pick up shit for you, so you shouldn't make this hard for them. You should bring all the materials to them, organize collection of samples, whatever is suitable for your particular situation. You should give them everything nicely prepared, nicely packaged, professionally designed, so they literally only have to bend over, pick up the sample, put it somewhere, and get it to a place where it gets to you. This is what you need them to do. Don't make them jump through hoops, because they're not going to do it, and then you're not going to get samples. What has worked really well for us is using swabs to collect samples. This is simple, it's clean, and it's much more convenient for people. If you make things convenient, then people are going to work, because it's not difficult. If you make it difficult, then people are not going to collect samples for you. And another thing, again, for anybody planning to do the citizen science stuff, one major thing that needs to be understood here is that volunteers don't work for free. They're just not paid with money. People have their reasons and their motivations they want to join your study. For some, it's the experience, the chance of doing something they think worthwhile, recognition, whatever. But you need to understand that. You need to acknowledge their participation. A T-shirt, project T-shirt goes a really long way. A certificate of collaboration or whatever, just to make to let them know that their work is appreciated. And another thing, you need to provide feedback, personal if possible. Otherwise, just through articles and so on. They need to understand and see the results of their work. You really need, if you take all this into account, if you work on that, you will have enthusiastic supporters for your next project, which is really important if this is considered to be a long-term monitoring program. If you ignore this, next time you're on your own. Maybe an anecdote. When we had meetings with hunters and foresters in this last study in 2023, many of the people showed at the meetings in T-shirts from the previous sampling. They were actually proud to have been part of the previous sampling. And this is what you want to achieve. This is what works. Okay. Maybe generally, to get back to wildlife monitoring, starting a monitoring program is difficult, it's expensive, it's a lot of work. But as this goes through monitoring cycles, it becomes easier and easier. As you learn how to do it, you learn the strings, you learn how to organize it, how to analyze stuff. On the other hand, the knowledge accumulates and management and conservation benefits accumulate. But it's really difficult to get over this first hurdle and actually start running this into a routine program. Also, practices get picked up if they work. Right now, we're starting, actually started a couple of months ago with a large project where we're extending this monitoring to the entire Daneric Pindos population. It's a large EU-funded life project, Dimpim Bear, where we're extending this into all the southern Balkan countries. On the other hand, we're right now analyzing samples from Bulgaria, where we helped design the sampling, and they actually implemented the sampling in the field and got the samples, and now we're doing the population size estimate also for Bulgaria. So if things work, people see, and then things expand, and then we can actually get to the point where the monitoring is done at the transboundary at the population level, which is what we're aiming for. So maybe to conclude, large carnivores are a very polarizing species. In Europe, we need coexistence to exist with them, and for them to exist with us, actually. Now, challenges to this coexistence come from both sides of the spectrum, from the people that love bears and would want to preserve every individual, and to the people that just can't stand them and would like to exterminate them. But coexistence will be by definition, a compromise. So both sides will need to give up something. And I would say that while advocating for education of bears certainly doesn't help their conservation, I would say that the same can be said for fanatical protection, which we also see of any individual, regardless of the cost. Bears do cause conflict with people, and sometimes we have to act also to protect the people. So if you're really trying to protect every individual, bear, wolf, whatever, that's not conservation, that's ego trip, and that really doesn't have place in conservation. So conservation is a compromise, but it must be a compromise done for the good of the species or landscapes we want to conserve. Okay, thank you. I hope you enjoyed the talk. These are my contacts if you have any questions, and bye.